Databases are a place where you can store your data, it's where you can submit it, it's where other scientists can access it, and then collectively all scientists can use it for their own research.
DNA Subway ties together bioinformatics tools and databases to assemble gene models, investigate genomes, work with phylogenetic trees, and analyze DNA barcodes. I'll work through an example to show you how to identify a species using a DNA barcode. This will help you to do the online activity. Search for DNA Subway in your web browser to bring up this page. You can register, or you can simply leave the username and password blank in the top left-hand corner and enter as a guest. Click on the blue square, Determine Sequence Relationships. The first thing we do is select our project type. In this case, it is barcoding with the CO1 gene. Then we upload our forward and reverse sequences. Click Browse and go to the folder where you saved your files. To select both files at once, hold the Shift key down on your keyboard, click on both file names and press Open. Then you can give your project a name. Click Continue. Now let's take a look at our sequences by clicking Sequence Viewer. Here you can see the unique combination of A's, C's, G's and T's in your forward and reverse sequence. You'll also see some N's at the end of the sequence. This tells us that the quality of the sequence was poor and can't be used in your analysis. To look at the trace files, click on the small icon. You can scroll from right to left, and you can change the scale by pressing the plus and the minus buttons. And you need to check that there is a nice clear peak in every position along your sequence. You will notice that the end of the sequence is very messy, and this can be trimmed off. We'll do that in the next step. Close this window, click Sequence Trimmer, click again. If you click on the icon, you will now see that the end of your sequence has been trimmed away. Next we'll do Pair Builder. Remember that we have a forward and a reverse sequence, but the computer doesn't know this, so you have to tell it which is the reverse sequence by clicking on the F. Then you can select both boxes, pair the sequences, yes, press Save. Close this window. And now we do the Consensus Builder. The Consensus Builder combines your forward and your reverse sequence to produce the one sequence which will be used in your final analysis. Click on Consensus Builder again. Here you see the forward sequence, the reverse sequence, and the bottom is the final sequence that will be used in your analysis. The dashes indicate that you don't have quality sequence in this position. Depending on your sequence, sometimes you may see that the forward and reverse sequence don't match, and so you can use the Trim Consensus feature to edit those positions. You can go back to your original trace files and see what is the correct read. You can also trim off the end of your consensus sequence if you like, if you feel that it is messy or may not be accurate enough for the analysis. You can click on the sequence and say what you would like to exclude. Finally, we click Blast N. Blast will basically compare your sequence to a database of other DNA barcodes and try to identify the mystery species. Click Blast. This may be very quick, or it may take a few minutes. Here you can see the length of the sequence, a score, and the number of mismatches. You can see here we have 0 mismatches, and this indicates that the mystery species is Helicoverpa zea. This is a species of bollworm. It is very difficult to identify species of bollworm using their physical features, but it is important to do so because some species are very destructive crop pests. See if you can identify the pests in the online activity.
Congratulations! So now you've learnt about lots of new things you can do in the lab, and you've learnt how to do DNA barcoding.